Okay, good to go?
Yep, good to go.
All right. So, okay. Good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you for joining us, the 28th seminar and University of Delaware Day, because both speaker today is from University of Delaware. So today also very special day for me because today is the first day when I am doing SynBYSS outside of St. Louis. And in fact, I am in the hotel room in Portugal. So this seminar series is really becoming a truly global event. And this is my first trip to Europe this year. And I will be traveling for probably more than 4 months to Europe 2 more times, Korea 2 times, Mexico, many US cities, etc. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Wilfred Chen. He is a Gold Professor of Chemical Engineering at University of Delaware and an editor-in-chief of Biochemical Engineering Journal. So that means he's a very powerful person. But more importantly, he is a big supporter for young people, including young faculty. So it is my great honor to have him here because a lot of young faculty respect him because his support for many decades. So it's kind of my great pleasure finally to have him today. So Professor Chen, the virtual podium is all yours. Thank you so much for joining us.
All right, thank you. Thank you for organizing this really exciting series. So today I'm just going to tell you a very short story. I think I've saw on the social media a lot of the either current PhD students or postdoc, particularly the first generation college kid, always a question: how do we navigate the academia landscape? So I personally is not only international, who moved here in the US when I was 16 years old from Hong Kong, but I'm also a true first generation college student. My parents only have a high school degree. And in fact, on my father's side, I don't believe anyone even have much of a formal education. They're mostly farmer in China. So another interesting thing is because I moved here when I was 16, I officially only finished 11th grade. So when I started to apply to college in the US, no one would accept me because to them, I never graduated from high school. So the only route for me to go is to go to our community college and hope to transfer to a 4-year college. So it took me about 2.5 years because I have to end up taking quite a few ESL classes. And I think Kevin probably heard of that story. During the meantime, I work at McDonald's flipping burgers for 2 years. So is my only industrial experience in the US. Very fortunately, I think able to transfer to UCLA to do my undergraduate in chemical engineering and really have no idea about graduate school at all. It was not until I worked as an undergraduate researcher in my junior year in the summer, one of the professors just saw me in the hallway and just grabbed me to his office and asked me, "What do you want to do?" And I told him, not sure. Wasn't really interested in working in industry because I didn't find it to be exciting. So he convinced me to go to graduate school. And that's how the whole thing started. So I would say my pathway to becoming an undergraduate in ChemE was just, I like chemistry, I like math. And as any Asian parents will tell you, you ought to be able to be an engineer to make more money. So that's how I become chemical engineers. So even after starting grad school at Caltech, finishing up a degree, looking for a faculty position was kind of an interesting experience too, right? You have no idea what to expect. And I did apply to a lot of places. And I have to admit, my interview skill back then was not good. So I ended up getting a job at UC Riverside, brand new college of engineering, brand new department, no graduate program until 1998 when I joined in 1994. So it was really interesting, right? You're joining a place, you expect to do research, but there's really no student, so to speak. I would say that I ended up learning a lot because you have to do everything from scratch, including writing the curriculum, getting ABET accreditation. But the more important thing I wanted to highlight to young people is the fact that you will never be able to do it by yourself because you need good internal mentor. You need a lot of really good external mentors. I mean, I think you mentioned Bill Bentley. I think Bill was one of the first person who sort of helped me. I remember talking to Bill and Mike Bittenbauer at one of the ACS meeting. They basically just told me, well, we're going to help you. Okay, so you really do need people like this from the outside to just sort of help you navigate this system. So as a result, I remember that, and I make sure whenever anyone who needs help, I'll make sure I will provide that kind of help from over the years. So I just hope that anyone who is a first generation doing PhD right now, finishing up, or doing a postdoc right now, if you're passionate about doing research, you think academia is the right thing to do, go for it, okay? I think the SynBio and the bio community is extremely friendly. And there are a lot of people here willing to help. And if you want any additional questions from me, feel free to email me. Happy to chat via Zoom anytime. So that's my short story.
Oh my God. Thank you so much. This is basically reflect your philosophy I felt for last decade. And thank you so much for your support for young people and faculty. And actually, that's my interesting story, your McDonald's story. So actually, I in fact got lost yesterday because I did not have any euro to pay for my parking fee. And then at the end of the crazily wandering for 2 hours, I found McDonald's, and I assume McDonald's should have some dollar to euro change, and also I assume McDonald's people should speak English, and because they are American franchise company, and however they do not speak English at all, but everyone in McDonald's in Portugal want to help me. Body language, whatever, and then I was so surprised. I mean, their kindness and so on. So basically, your story is all about kindness and then caring about the people. And I'm very touched by your story. So thank you so much for that one. That's very inspirational, and I'm really glad and honored to be host of your seminar and the Kevin seminar today. So thank you so much. I mean, I'm delighted to have you. So thank you. Okay. So now the main speaker of today with a longer introduction. Dr. Kevin Solomon is an assistant professor of chemical and biomolecular engineering at the University of Delaware, the same department of today's pioneer speaker, Wilfred Chen. He holds a bachelor degree in chemical engineering and bioengineering from McMaster University in Canada, and master in chemical engineering practice from MIT, and PhD in chemical engineering from MIT, actually in Chris Prader lab. His work focused on identifying and engineering environmental microbes and microbiomes that are well adapted for applications in sustainability, materials, and health. And he has received numerous awards, including the US Department of Energy Early Career Award in 2019 and an Outstanding Faculty Award from Purdue Residencies in 2018. And recently, just this year, he received NSF Career Award, another very competitive one. So congrats, Kevin. This is wonderful. And then I believe that you are true rising star. He also has provided expert testimony before the 116th US House of Representatives on the convergence of engineering and biology, and has co-authored technology roadmaps for engineering biology. He has also been featured in Forbes magazine and was recognized as being among the 1,000 inspiring Black scientists to watch. Dr. Solomon's work is supported by NSF, DOE, USDA, and NIH, private trust, and industry. So he's really rising star. And if among audience, there are big names writing career or tenure letter for him, please pay attention to his talk today. Kevin, I'm not giving you pressure again, but I'm talking about your, big and great work. And I'm looking forward to listening to your talk today. And Kevin, and take it away. It's all yours now. Thank you.
Thank you so much for the introduction. It's always great to be in the presence of such greats, actually, both you and Wilfred. It's always great to hear great advice and to reassure me that I'm not totally crazy and going too far off the beaten track. Can you guys see my slides right now?
Yes.
All right. So for today's talk, I just wanted to really highlight some of our efforts in domesticating environmental microbes, as you heard, for different applications. But before I go into that, I just wanted to take a moment to acknowledge the hard work that I'll be talking about today. This is not inclusive of my entire lab, but I just wanted to acknowledge Ethan Hillman, one of my first PhD graduates, who's now a postdoc at Michigan Med School. Casey Hooker, Ethan Hillman, Casey Hooker, a postdoc, Rado, who's joined the team, an exceptional undergraduate, Adrian Ortiz-Velez, and 2 rotation students who joined my lab. And so you'll see their faces pop up occasionally because they're the ones who really did all the hard work, as you'll hopefully appreciate. And again, I also want to acknowledge the Department of Energy and NSF, as well as our collaborators at a variety of institutes for their support. So why do we do what we do? So hopefully many of you recognize that as a society, we're addicted to carbon, right? I mean, the current crisis in Ukraine probably has brought it first and foremost in discussing the difficulties that Europe has in weaning itself of Russian oil. And there's been a lot of discussion about, for example, the price of gasoline, liquefied petroleum, so among other things. And so there's a lot of discussion about the energy content in a barrel of oil and how difficult it is to replace it. But ultimately, those sources of energy can potentially be supplemented or replaced by alternative forms such as nuclear, solar, wind, et cetera. However, there's far less discussion about this 18% that ends up in other products. And that 18% represents physical carbon that actually goes into things that we need, right? And so that is quite substantial, right? I mean, we're talking about the carbon that ends up in the performance polymers, that ends up in fibers such as our clothing, carpets, and among other things. The carbon that ends up in our medicines, the carbon that ends up in performance materials such as polymeric coatings that keep our iPhones from overheating, that keep them resistant, and so we don't get electrocuted. and even our foods. So things such as gummy bears have carnauba. So we're not talking about the energy content to make like fertilizer. We're talking about a barrel of oil is directly in the food that we consume. And so we live in a fundamentally carbon-based society, and that carbon right now is derived from oil. And as we think about going forward in the future, as oil becomes more expensive to access, as we think about oil being localized to isolated areas where there's a lot of conflict, among other things, there's a significant push for alternatives. Now, fortunately for us, biology can fill that gap, right? So that the classical example are plants. They're able to fix atmospheric carbon dioxide to make cellulosic polymers, which are just complex, a number of different sugar molecules that then a number of microbes, either naturally or engineered, can then ferment into a wide array of products. And there's a large infrastructure, a wealth of development in synthetic biology, in metabolic engineering, chemical engineering, bioprocessing to make this happen. And so, like, what are the types of things that we can make, right? So, I mean, microbes, they're very good at chemistry, and just on their own, they will ferment these sugars into things such as acetone, butanol, acetate, ethanol. And up until World War II, microbial processes, so the ABE process, that was the dominant way by which we could make some of these compounds. And this was ultimately how the British made certain explosives such as cordite. They actually used Clostridia to make a lot of these compounds. If you look at other microorganisms, so you look at fungi in particular, you will see that they have larger molecules that actually are bioactive, and these are the basis of many medicines, such as lovastatin, a blockbuster anti-cholesterol drug, erythromycin, an antibiotic. And we're now getting clever to the point that we're recognizing that even common molecules that seem to have very low value, such as, for example, this amino acid, it's a decarboxylation and a deamination away from a traditional commodity chemical such as styrene. And so when you look at the sum total of metabolism, the sum total of chemistry that life is capable of, you get this very complicated map where these different nodes represent different compounds and the lines represent different enzymes. And as I said, if you're a clever engineer or a synthetic biologist, you might recognize that, for example, these terpenoids or polyketides, which are bioactive, they're cell defense compounds, they're the basis of many pharmaceuticals, amino acids, commodity chemicals, lipids for biofuels, and so on and so forth. Now, there's been a very significant effort in trying to engineer microbes to access different parts of this map. But what we're beginning to come up against is the fact that some of these chemistries, they just require exotic conditions. So, for example, they require more reducing conditions. They require environments where that have specific ratios of cofactors to fold enzymes correctly, which aren't accessible in the traditional synthetic biology chassis. And so to bridge that gap, my lab is very much interested in onboarding or domesticating environmental microbes that access these interesting but hard to reach parts of this metabolic map. Now, to do that, we use systems biology approaches. So we use genomics, transcriptomics, proteomics. We use all the omics to study and characterize these interesting microbes that may or may not have interesting capabilities for biocatalysis. We use bioinformatics techniques to really dig in to understand what these organisms are capable of, what enzymes they have, what are the strategies they use to do this. And then, of course, we use synthetic biology. So we then think about how do we take established tools that work well for E. coli and yeast and other organisms? How do we, for example, implement CRISPR in this organism that was locked in a deep sea vent 5 years ago? How do we onboard and bring these technologies on? And in many cases, it turns out that the tools that we have just don't work. And so there are some fundamental efforts in my lab developing novel gene editing tools, coming up with new ways of engineering protein activity, among other things. And so a particular focus in my lab is this challenge of sustainability, right? And how are we able to replace the carbon that we're currently pulling from a barrel of oil with waste carbon, and in particular, a waste such as plastics or abundant renewable plant biomass. So the parts of the plant that we don't eat, the parts that end up rotting in fields or getting burnt for fuel. How can we capture that waste carbon in it? And it turns out that if we look at animal microbiomes, their gut microbiomes have evolution has already solved these problems, and they're interesting microbes in these isolated environments that are able to actually degrade these things. And so, for example, we've found, and we're currently studying in collaboration with the Blenner Lab, looking at the capacity of different mealworms or insect larvae to degrade post-consumer plastics such as polystyrene, polyethylene, polypropylene, as well as we were looking at the ability of other gut microbes from large herbivores to degrade lignocellulose. Now, in the interest of time, I can't really talk about all our efforts. And so for today, I want to focus on our work to domesticate and to bring on board these anaerobic fungi. But before I go into the synthetic biology of how we're engineering these things, I want to take a step back and talk about what are their properties and why we use them. So what is the fundamental challenge that we're trying to address here? As I said, plant material, the parts that are non-food-like, they are a vast reservoir for trapped carbon in the form of polymerized sugars such as glucose, arabinose, and xylose, which can be fermented by a wide array of microorganisms to produce value-added products that we can engineer. And that glucose is, for example, trapped in the form of cellulose. The pentose sugars and the hexose sugars are locked in the surrounding layer of hemicellulose, and then this whole thing is encased in a lignin-rich or a heterophenolic polymer layer. And so it turns out that to get access to these sugars, we really need to break open this thing. So we use pressure cookers, we use high acids and bases and high temperatures to really break open, get access to these inner layers, and that's very energy intensive and inefficient. And then we take enzymes that we pull from a variety of different microorganisms that can then depolymerize these, get free sugars. However, the conditions that we currently use to break open these plant materials, so these high temperatures, acids, and et cetera, they are denaturing to the enzymes that we ultimately use to depolymerize it. And so they have very short half-life. A very short half-life, and because of this, they're very expensive to produce because we need to make a lot of them. And then more importantly, as we think about different types of bioenergy crops, like what you might find in California versus Missouri versus in Delaware, those are going to be different bioenergy crops, very different ratios of these plant wall components, and therefore very different cocktails of enzymes that you need to depolymerize different things. And so there's no one-size-fits-all strategy, and it's very inefficient. However, if we look to natural systems, so look to large herbivores such as cattle, horses, goats, giraffes, et cetera, they're able to survive on these things. And it all comes down to the activity of these little tiny things here. These are anaerobic fungi. So they're fungi that are uniquely anaerobic. And they're able to degrade a wide range of plant material, and in fact, degrade up to 60% of all the lignocellulose that's ingested by these animals. So for those of you who are wondering what are anaerobic fungi, just to kind of give you a quick primer, this is a fungal tree of life, later diverging strains at the top, early diverging at the bottom. And so at the top, you have your multicellular things such as mushrooms. In the middle, you have your things such as your yeast. These early diverging Neocallimastigomycota are anaerobic fungi. And unfortunately for us, there are no synthetic biology tools. Genomes are AT-rich, which I'll elaborate on what that means in a second, in a couple of minutes, as well as we have to actually, there are no model strains. We can't order it from ATCC. We have to actually go out in the wild and get it ourselves. And so when I started my independent career, I was based at Purdue University, which is in the Midwest in Indiana. And fortunately for us, there are lots of local farms. There was an Indianapolis Zoo. And so we went to these local farms and we collected samples from these large herbivores. So you can see we had actually a miniature pony, some goats, among other things. you collect these fecal samples, and then using classical microbiological techniques, we can actually isolate single colonies of pure species that we can then grow and cultivate and characterize. And you can see here, here are the first three isolates in my lab. You can see that they come from two different genera. They have very different morphologies, and they all have different capabilities in terms of their ability to degrade lignocellulose. However, what was interesting to us was that they were able to grow robustly on a wide range of plant materials. And so here, because these things are anaerobic and because they grow on particulate substrates, the way that we track growth is by tracking the accumulated pressures of the evolution of CO2 and H2 gas as they grow. And you can see here, if we've normalized everything to growth on glucose, as you feed them complex plant biomass, such as alfalfa, corn stover, sorghum, wild-type poplar, you can see the growth on these complex materials is on par with that on pure glucose, which suggests that depolymerization of these complex materials is not rate limiting for growth, i.e. they're very good at breaking down these materials. And you can see that even in some cases, they actually prefer these types of materials. And so we were able to extract and isolate secretomes and isolate the different activity of the different enzymes were available. We've done zymograms, so you essentially overlay your page gel with substrate, and you can actually get clearing zones, which tell us what the function of individual proteins in the secretome are, among other things. And one of the more intriguing properties that we noticed was their ability to overcome S-lignin. So what do I mean by that? So recall that I said that plant material, it has this heteropolymeric, heterophenolic polymer called lignin that surrounds the entire thing. And that lignin is actually composed of three different subunits, H, S, and G lignin, which differ in the amount of cross-linking they can do through these methoxy side chains. And many fungi, including those that are actively lignin degrading, they have a very strong their rate of hydrolysis of these polymers is very dependent on the specific composition. And so as you have higher levels of S-lignin, degradation is much slower than if you have low S-lignin. And that's just because there's more cross-linking in these structures. However, with our anaerobic fungi, not only do they grow at a comparable rate across a wide range of S-lignin, if we look at the total sugar release, and this was over a week period, I believe, if we go from 5% to 95% S-lignin, you can see that the total amount of sugar release is comparable across all strains, suggesting that this is not a bottleneck for these organisms. And we can actually see that they express a number of different enzymes as a function of S-lignin to overcome this challenge. And so what we're discovering is that if we look at the genomes of these organisms and annotate their capacity to express enzymes that degrade these materials, we can see that the anaerobic fungi have significantly more enzymes than current industry standards such as trichoderma or aspergillus, which suggests that this is a more robust and more viable platform for biomass degradation. As well as it has this unique strategy in that it has an extracellular cellulosome, where they put many enzymes that degrade plant biomass in close proximity. These enzymes have different functionality, but by putting them in close proximity, they work synergistically to hand off the material from one enzyme to the other, ultimately giving you far higher hydrolysis than if you had them in isolation. And so we think that these two key properties are what drives this efficient hydrolysis from these organisms on different substrates. So since we can use these things for plant biomass degradation, people have asked the question, well, can you use this as a way to liberate free carbon that you can then feed to another organism to make chemicals or fuels? So can you partner this with something such as E. coli or Saccharomyces that we know how to engineer well and turn this into products? And while people have been successful and they've done this to get decent titers of ethanol, what we found is that to improve the efficiency of these processes, you want them to go longer, right? So you want to increase the overall hydrolysis efficiency. And as you increase the hydrolysis efficiency, you do get more sugars, more glucose, xylose, and arabinose that can be fermented. But the challenge here is that the fungi actually then actively eat a lot of that extra sugar and make organic acids such as lactate, formate, and acetate. And so there's this tension between overall process yield as well as efficiency, because if you want to get more carbon out, there's going to be some loss of that carbon from free sugars to organic acids. And so the question is, could we improve the overall efficiency by actually reusing those organic acids as well? And just to kind of give you a sense for the scope of the problem, here I'm showing you the hydrolysis of different substrates. So SynBYSS is pure cellulose, corn stover, poplar, and alfalfa. These are untreated plant biomasses. And you can see that our fungi are able to degrade at least 70% of this material without any issues. However, the vast majority of the carbon that's liberated is not free sugar such as glucose, xylose, or arabinose. A lot of it is organic acids in the form of of lactate, formate, et cetera. And so we decided to partner this rather than E. coli or Saccharomyces. We decided to partner this with another emerging non-model fungi. In this case, the yeast K. marxianus, which is the fastest growing eukaryote and grows at high temperatures. And we found that it was able to utilize the vast majority of the carbon with the exception of formate. And in using this, it actually grew quite rapidly and grew on par with what you see from traditional YPD media. And you can see that the tighter, that the overall biomass formation is quite high. We saw ODs of roughly about 30 over 48 hours, and this is comparable across all substrates, regardless of what it was actually made of. And so we then asked the question, what can we make with this? And so one of the other beauties of K. marxianus is that it's very good at making esters, among other things. And so these sugars and organic acids were fed to this. And we found that when we compared the concentration of esters such as isoamyl acetate, if we isoamyl acetate and isoamyl alcohol, if we compare the yields on YPD versus plant biomass, which has been degraded by our fungi, we get titers and ultimately yields that are comparable across all these things. And so this is one of the first demonstrations of going directly from plant biomass to these high value solvents and flavors without any traditional bioprocessing. And then, of course, with traditional synthetic biology, so for example, by knocking out this alcohol transferase, we can reduce titers of that ester and make exclusively the alcohol. And again, we have comparable yields. And we see that for other products, such as, for example, ethyl acetate, which is a solvent in fragrance, and even other aromatic alcohols, such as 2-phenylethanol, we can get very high titers from the system with, again, very high yields going directly from plant biomass. So now that I've kind of convinced you that hopefully convinced you that these environmental organisms are really powerful for biotechnology, how do we actually engineer or control them? How do we implement systems and how do we implement synthetic biology in these organisms? Well, recall that these things were things that we pulled from a horse, from a donkey, from a giraffe about less than 5 years ago. So in the span of 5 years, so literally we are back in the days of E. coli research in like the early 1920s, 1930s. We have an isolate, but we really have no idea what to do. And so we have to start from scratch. We need a genome, we need to figure out how to get things into the cell, we need to figure out how to engineer them, et cetera. And so one of the first things that we need to do is to get this genomic map. And for us, one of the key challenges was that we needed to have a complete genome because we needed to develop parts. Like we needed to know what was a promoter, and you can pull a promoter from context based on the fact that it's upstream of a coding region. Now, I alluded to the fact that these genomes, these organisms, they have quite low GC content. So their genomes are about 15% GC. So that means they're 85% AT. And for those of you who do any sequencing, next generation sequencing platforms such as Illumina, they have far lower accuracy for AT-rich regions. And more importantly, because they're AT rich, it implies that there are regions where there's a lot of homopolymeric repeats. And so when you try to reassemble your sequencing reads, there's not enough, there's too much ambiguity to have a complete genome. And so what we have is very fragmented. And so in the last 10 years, genomes have gone down from about 30,000 fragments to down to maybe a couple hundred, but that's still far away from having a complete closed genome. And so to address this, we leveraged a newer technique called high-C or chromatin conformation capture sequencing, where essentially before we do our genomic sequencing, we cross-link our DNA and make a library that essentially preserves the interactions between DNA pieces. And you would expect that DNA that is next to each other, so DNA that should be assembled next to each other, will have higher degrees of contact. And they will be read together at the same coverage depth and frequency due to this cross-linking. And so based on this technique, we can make these contact maps of pieces of DNA versus itself. Things that are more red have contact enrichment, things that are white have no contacts. And you can see based on the assemblies using even long-read pack biotechnology that you get a lot of off-diagonal contacts, so things that are not next to each other, which tends to be inaccurate. So it suggests that the best assemblies using traditional pipelines was inaccurate. However, once we leveraged that contact data, we were able to then to fix some of these misassemblies, rearrange our sequences, and actually get chromosomal resolution in these things. And so we're able to get more high fidelity genomes. And so just to kind of give you a quick snapshot of what that means, if we look at the size of the percentage of the genome versus a scaffold or the contig, the fragment size, you can see that just PacBio alone or unphased, they give you very small pieces. However, our phased genome, there's a significant increase in the fragment size to what we think are actual chromosomes. And actually, if we look at the statistics, we can see that we've improved our N50. So higher N50 is better, is reflective of a better assembly. We're able to correct hundreds of errors in the previous assemblies using conventional technologies. And more importantly, we're able to get our genome down to what we think are actual chromosomes. So we can see that these different isolates, they have between 12 and 24 chromosomes. So now that we have these genomes, we now have this deep reservoir for parts that we can mine. We have to figure out how do we even get our DNA into our organisms. Now, fortunately for us, these organisms, there's a part of their life cycle. So these are fungi, so they go through multiple stages. However, in this immature or juvenile zoospore life cycle stage, they're naturally competent for nucleic acids. And so if you mix them with DNA or RNA, and we've on the order of about a microgram or so, you can see that they get internalized and they get localized to the nuclei quite readily. And so we've leveraged this along with our ability to, with our growing part database of constitutive promoters, among other things, to begin to develop a full suite of tools to enable engineering. And so, for example, we've characterized and tested a number of selectable markers. We've demonstrated that we can actually introduce them and rescue growth. So for example, here's a negative and positive control. Our fungi, when we use an antibiotic such as hygromycin, they don't die, but we can rescue that with our selectable markers and our promoters that we have. We have enzyme reporters, fluorescent reporters, among other things, and we have now a burgeoning toolbox. The one thing that we do currently lack, for example, however, is we don't have a way of stable transformation. So we still don't know what a plasmid looks like in these species. All right. The way that we're achieving these phenotypes is by dosing in our plasmid at multiple intervals, and that's because we can, because they're naturally competent. However, these pieces of DNA are not replicating, which is why we need to dose them in multiple times. And so we're now thinking about how do we implement genome editing technologies such as CRISPR or some other alternative platforms that my lab is developing so that we can get these stable phenotypes. And so for that, we've identified NLS tags and other tools that will allow us to not only express proteins in these cells, but to target them to intracellular compartments. So the NLS tag will allow us to potentially express Cas9 or CPF1, among other things, in the nucleus, so that we can then think about how to engineer these things. As an alternative strategy, we're also beginning to look at chromatin structure or the epigenetics of these anaerobic fungi. Now, just as a quick review for people who don't think about chromatin structure, in fungi, like all eukaryotes, their DNA is wrapped around histone proteins. And those the spacing between those histone proteins essentially controls how accessible the underlying DNA is to the transcriptional machinery, and therefore whether a protein is expressed or not. Now, these histone proteins can be post-translationally modified, so they can be methylated, acetylated, among other things, and by a variety of different enzymes, and that activity will ultimately control the spacing of these things. And so, for example, H3K4 trimethylation is associated with expanded chromatin or euchromatin, which leads to gene expression. H3K27 trimethylation is associated with condensed or heterochromatin, which represses or silences genes. And it's been shown that a number of environmental stresses, such as temperature, pH, and microbial interactions, or even small molecules, they act as inhibitors for these different enzymes. So, for example, a histone deacetylase, which removes acetyl groups from these histones and catalyzes a transformation from one form to the other. And so we asked the question, how powerful are these strategies in anaerobic fungi? And so we validated that, for example, an inhibitor such as SAHA, here I'm showing you positive and negative controls for these marks and these antibodies with Saccharomyces cerevisiae. But the key thing to note is that our anaerobic fungi, under laboratory conditions, they show very low H3K4 trimethylation and high H3K27 trimethylation. So both of these marks in this configuration are associated with gene repression. However, by supplementing them with this chemical compound, we can do a global switch to a more expressive condition. And we see that when we do that, we actually get a twofold increase in the amount of xylanase activity. So there's an enhancement in the ability to degrade lignocellulose. Similarly, if we do a survey with other inhibitors such as valproic acid, nicotinamide, butyrate, we see a number of changes on the order of 2 to 4-fold differences. And this comes with a regulation of hundreds of different genes as a function of these things. Now, while we're still processing this data and trying to understand exactly how these phenotypes come about and how best to modulate them, we have noted that, for example, butyrate, it does seem to induce a substrate specialization. And so lignocellulose is made up of cellulose, so glucose, and hemicellulose, which is glucose, xylose, and arabinose. And butyrate, which is a metabolic end product, it tells the fungi to leave the other parts alone so that the other microorganisms which are producing butyrate, such as bacteria, can have something to eat. And so it seems that these epigenetic strategies are not only powerful in controlling gene expression and modulating these phenotypes, but they're important in mediating cell-cell communication in these microbial communities, and we're beginning to probe into this. And so just to wrap up really quickly, hopefully I've convinced you that anaerobic fungi are a great platform for biomanufacturing, primarily because they have a very large and robust repertoire of enzymes to degrade plant biomass. These enzymes can overcome particularly recalcitrant biomass components, which opens them up to accessing a wider range of plant materials. We can right now partner these organisms with other engineered microbes to directly upcycle this waste carbon to other things. We now have the beginnings of a genetic toolbox that will allow us to do synthetic biology in these organisms and directly engineer them going forward, as well as we've also demonstrated that we can use epigenetic strategies such as chemical modulation to control activity. Now, before I wrap up, I just want to do a quick plug. so some of you may or may not be aware of this journal, Synthetic Consistence Biotechnology. It's one of the newer SynBio journals, but in the last couple of years, it's had a very high impact factor of roughly about 5. Myself and Professor Sandoval at Tulane University, we are guest editing a special edition on engineering gene regulation. And so if you have anything in any of these topics, so transcription factor engineering, CRISPR-IA, cell-free prototyping, modeling, or anything in between, we are accepting submissions through August 31st. So please reach out and let us know if there's anything that you have in the pipeline that would be of value. At this time, I would just like to thank you all for your attention and thank again our sponsors, the contributions of many people in my lab, both former and present, and I can see that a couple of them are in the audience. And thank you all for your attention. Happy to take questions.
Okay, wonderful. Thank you so much. Amazing talk as always. So let me see any question from chat or Q&A. In the meantime, if Wilfred, do you have any comment or question?
I think Udell is very lucky to be able to steal Kevin from Purdue. I think he's going to define our SynBio area with Mark and Aditya for the next 20 years to come. So,
wow, that's a lot. Great compliment. So, actually, I failed to steal Kevin from Purdue and from you, unfortunately. So, yeah, that's our fault in a sense. Or okay. So let me check Q&A. This is not the question actually. Chat. Yes. No. Okay. Let me start the one question. So Kevin, interestingly, I chat with one of the industry guy today, not during the conference, but actually another conference call. I need to do that. So about the plastic recycling or upcycling, because you are doing something, but you probably didn't cover today much, but I just want to hear from you about your vision or perspective. The person I talked to was basically chemistry person, and then using some, traditional metal, catalysis and so on. And then what he said, or even not talking about even chemical recycling, the physical recycling might be sufficient enough to kind of solve some problem of the plastic problem. And of course, my point of view is basically the biological process much better in terms of improve the value out of this one. And also he also adds some chemical, recycling also another alternative, but there are some, debate about that one. So I just wanted to hear from you about what's your thought and what's your perspective. And I wanted to hear from you.
Okay, so I think so. So I'm trying to choose my words because I don't want the catalysis people to freak out and get upset with me. But so my understanding is that I think that there will be room for biology for the simple fact being that most waste plastic, so post-consumer waste plastic is not clean, right? Mm-hmm. So I think if you have virgin plastic, so if you have like leftover scrap plastic from an industrial process, it's really easy to just reprocess it. Or if you want to upcycle it, there are tons of catalysts that will do that. The challenge becomes just like, for example, when you have your takeout container, like your styrofoam container, there's like all the oils from your Chinese food and all these other things. And those things, when you put them through a traditional and catalytic process, they essentially poison the catalyst. You get a whole bunch of undesirable side products. And so the beauty about biology is that they naturally filter and screen out a lot of those impurities. And so even though you might so that you can still process the same plastics, but they don't have to be clean, which is one of the huge economic barriers, trying to sort out your good plastic from the bad plastic and cleaning it. And so from that perspective, I think there will be room for biology. Now, what we've found is that even though they're not overly common, there are tons of microorganisms that seem to love plastics. And so, for example, we've focused on the mealworm digestive tract. And if you're wondering why evolutionarily, why they even eat plastics in the first place, insect larvae, they eat leaves. These leaves, they typically have their cuticles, so the coating on the leaves, it's a waxy component which they use to keep the leaves waterproof and to keep it from evaporating and losing all its water. And those waxes look very much like plastic. And so they have a large diversity of enzymes that can actually access these materials. The only challenge is that these organisms, they live in places that aren't readily accessible. And so what we envision is not only identifying these enzymes, but building artificial consortia that you can then put into a bioreactor or an anaerobic digester or some kind of industrial bioreactor of some kind that would then ultimately process these materials. The question then becomes how genetically tractable are these organisms? How easily are they to then engineer and manipulate them so you can then directly control the product portfolio of what they make? And so right now we have a current project which is looking at the first half, how do they degrade these materials? But actually we're about to pull the trigger and submit another proposal today talking about how to do the second part. How do we engineer these weird bugs that aren't E. coli, so don't have tools, don't have plasmids that you can readily plug in and go? How do we engineer them so that we can actually make what we want? So, I mean, I think it'll be a long road, but the long and short of it, I think in the next 5 to 10 years, you'll hear interesting stories about full bioprocesses that can take waste plastics that have all sorts of garbage on it and still make valuable products out of it.
Wonderful. Actually, now I confirm my answer is supported by you. So that's I'm glad I asked that question, because the similar answer I gave to my partner or my potential collaborator. So my argument probably valid and supported at least from you. Okay, wonderful. Mm-hmm. So I have the one question from chat. Said wonderful talk and really exciting work. I completely agree about the S-lignin and its influence. And the question is, does the S-G ratio matter? And that's the question.
Yeah, so I'm sure. So the short answer to that is I'm sure it does, but I don't currently know exactly how. And so I kind of hand waved and didn't go into the details about that study, but we increased the S-lignin percentage by reducing the G-lignin percentage. And so that S to G ratio wasn't independently controlled. And so I guess indirectly we're seeing that it matters, but I couldn't tell you what the specific impact of that ratio versus just having far more S-lignin is.
Okay, and then yeah. So that's Pri and said awesome, thank you. And I saw one hand by Armando, but I sent message, but he didn't respond to me yet. So I just wait a little bit more. So Wilfred, do you have any comment or last minute question? Otherwise, I probably close for today. And actually, I want to check out the beach outside. Okay, so I want to close, but we'll chat. After this one. So again, amazing talk, Kevin. Thank you so much for your wonderful talk as always, and also thank all for joining and staying today. We'll meet again next week on April 14, Thursday, the same time, the same Zoom link, but I'll be back to St. Louis on that Thursday. We'll have Professor Chris Voigt at MIT, my postdoc advisor, and Dr. Ruben Uribe at Technical University of Denmark. And actually, I will be visiting them. As usual, the follow-up informal chat will occur without recording. And so please stay here if you are interested in chatting with today's speakers and me, and I'll promote you to panelists who can speak and show your handsome and pretty face. And also, I can show my room and the ocean if this internet can allow me to do that. So I will stop recording and thank you everybody. And I stop recording. Stop.